Welcome to Plant Breeding of the Future. Figure 3 describes key microRNA mediated regulatory modules involved in 7 main stages of plant development in cereals: grain filling, floral organ development, floral transition, reproductive branching or spikelet formation, leaf morphology, vegetative tillering, otherwise known as the formation of additional stems, and lateral root development. Of course, all of these can potentially be engineered to reach certain breeding targets. For example, to improve photosynthetic capacity and the ability for a plant to cope with abiotic stress, targets may include leaf size and shape, or nutrient and water uptake, or even tiller number. If impacting the end product, however, that is the grain, then breeding targets might well include early flowering, male sterility and fruit size, spikelet size and grain number, and of course, grain size and quality.
The most well-characterized microRNAs known to affect flowering time are miR-156 and miR-172. miR-156 targets squamosal promoter binding protein-like genes, known as SPLs. SPLs are transcription factors well known to positively regulate grain filling, time of flowering, and reproductive branching, as indicated by showing these numbers from the panel in A and in blue circles. SPLs also negatively regulate lateral root development and tillering, as indicated by the orange number circles. However, miR-156 inhibits miR-172 expression, while SPLs upregulate miR-172, which targets apoptosis-2 or AP2, a repressor of flower transition. Completing the circuit, AP2 also inhibits miR-172 and upregulates miR-156. miR-529 also affects SPLs and therefore reproductive branching and tillering.
In yet another regulatory circuit involving 4 microRNAs, miR-159 targets MIB transcription factors, which negatively regulate floral organ development, as does TCP, which is targeted by miR-319. TCPs also negatively regulate leaf size. Both miR-319 and miR-159 induce miR-167 expression. miR-167 targets auxin response factors, which positively regulate floral development and lateral root development. In addition, TCP induces miR-396, which targets growth regulating factors or GRFs. GRFs positively regulate reproductive branching and leaf morphology. The regulatory circuit is completed when GRFs activate the ARFs. A number of other genes targeted by microRNAs also negatively regulate reproductive traits. These include universal transcription factor, nuclear factor Y subunit A or NFYAs, the circadian gene TOC1 or time of chlorophyll A and B expression, the brazinosteroid sensitivity related gene lacase-like gene LAC, and the auxin receptor transport inhibitor response 1 or TIR1. The NAC transcription factor targeted by miR-164 positively regulates floral organ development. The NFYAs also positively regulate lateral root development, which is negatively regulated by TIR1 and the repressor of the ARFs, IAA28. TIR1 also negatively regulates vegetative tillering.
In summary, each of the microRNA target modules that have been shown here could be used in smarter, de-stressed cereal breeding in the future.